the acid-fast stain using the Zeil-Nielsen method. To begin the acid-fast stain, you must first prepare your slide. Using one glass slide, label it Acid Fast. Draw 3 circles on the slide. One circle will be used for a positive control, another for a negative control, and a mix of the 2 bacteria will be placed in the middle. After labeling your slide, flip it upside down so the labels are on the bottom. You do not want the ink to get washed into your bacteria during staining. Now, on a clean slide, place some clean water. Distilled or deionized water is fine. Using a sterile loop, you will transfer 1 loopful of water into each of the circles on the labeled slide. The water will be used to mix your bacteria onto the slide. Now that you have finished adding the water to each of the circles on the slide, you are ready to start adding bacteria. Here, Staphylococcus aureus is being added first as a negative control. Mix the bacteria carefully and gently with the water in the circle labeled negative. Then, carry the loop over and mix it in with the water in the circle labeled mix. This process will be repeated after sterilizing your loop with the positive control. We will be using Mycobacterium smegmatis for our positive control. Mycobacterium is grown on Lowenstein-Jensen agar. This is a sterile egg yolk agar. Mycobacterium tends to be very waxy and sticky and can sometimes be a bit difficult to get off of the agar. You may need to use a little bit of pressure with your loop on the agar to obtain enough Mycobacterium for staining. Now, the Mycobacterium will be added to the positive labeled control circle first. Once the Mycobacterium has been mixed in that circle, the loop is then carried over to the mix and mixed with the Staphylococcus. After placing the bacteria on the slide, allow it to air dry. Once the slide has air dried, it should be heat fixed. After heat fixing, in the Zeil-Nielsen method, moist heat will be applied. Here, the slide is put over a beaker of steaming water on a hot plate. The steam will loosen up the waxy outer coating of the mycolic acid in the peptidoglycan of the cell wall. This will allow greater penetration of the phenol-containing stain or primary stain known as carbonyl fusion. A small piece of paper towel has been cut out that is about the size of the actual slide itself. The paper towel is placed down on top of the slide, and then the primary stain, carbonyl fusion, will be added. The paper towel serves 2 purposes. One is to prevent the stain from running off of the slide during steaming. The second purpose of the paper towel is to prevent the stain from drying out due to the steaming. The slide will remain on over the steaming water for approximately 5 to 7 minutes. It is important to observe the staining process during this time to prevent the slide from drying out. When the stain begins to dry out on the paper towel, this greenish metallic color will be present. A few drops of carbonyl fusion should be added at this time. The slide should be checked every 1 to 2 minutes. For acid-fast staining, the Zeil-Nielsen method requires the steaming step. Another acid-fast stain method, known as the Kinyoun method, does not require steaming. However, the carbonyl fusion contains a higher concentration of phenol. Once the steaming step has been completed, the slide is removed and placed onto a staining tray. At this time, after allowing the slide to cool for 1 minute, remove the piece of paper towel. After removing the piece of paper towel, place it into the staining tray, and you are now ready for decolorization. The decolorization step in acid-fast staining uses acid alcohol. The acid alcohol is gently run down the slide. It is helpful to rinse both the front and the back of the slide. When you rinse the back of the slide, your labels will be rinsed off. When decolorizing, you want to run the acid alcohol over the slide until the alcohol running off of the slide becomes a very light or clear purple. Over-decolorization will cause the mycobacterium to have too much of a blue color and be difficult to see. Too little decolorization will cause the Staphylococcus aureus to obtain a purple color, making it difficult to differentiate it from the actual mycobacterium. After decolorization, do a quick water rinse and then apply your counterstain. The counterstain is methylene blue. Allow the methylene blue to sit on the slide for 1 to 2 minutes. Carefully rinse the methylene blue with water. After rinsing, the slide is now ready for drying. Bibulous paper is used. The book of bibulous paper is opened, the slide placed in, and the book is actually tapped or gently rubbed to dry the slide. Do not wipe the slide with a paper towel. After drying, the slide is now ready for observation under the microscope. Acid-fast positive cells will turn a bright fuchsia color, and acid-fast negative cells will turn a bright blue color. In this slide, Staphylococcus aureus is acid-fast negative. Mycobacterium smegmatis is acid-fast positive. The bright blue background of acid-fast negative cells gives strong contrast for the observation of acid-fast positive. The acid-fast stain is an important tool in diagnostics, especially for the diagnosis of tuberculosis. The acid-fast stain is a simple and easy method to determine the presence of mycobacterium or mycolic acid-containing bacteria.